Welcome, everyone, to our first pitch session of the day featuring 5 companies within the Therapeutics sector. This session is sponsored by Genome BC, who has been a valued partner of our Invest in BC conference for many years. Just a reminder to post any questions you have in the Q&A section on the right-hand side of the screen. The presenting company guide will be posted in the chat area shortly as well. We will not have Q&A through the pitches, but our innovators will be monitoring that and will get in touch with you once they've finished their presentation. So to start, I would like to introduce Dr. Brock Schumann, CEO, Azor Biotech. Brock.
Great, thank you, Wendy, and thank you to the Life Science Selection Committee for including me. I am undoubtedly one of the earliest stage ventures presenting today, and I think it speaks to some promise they see in our technology that we've been included. So Azor Biotech is engaged in RNA-guided drug design and development, which is to say RNA-compatible, structure-guided drug design and development, going for the holy grail of unlocking the utility of RNA drug targets by tailor-making therapeutics that are small molecules. And these small molecules are important because oligonucleotide-based therapeutics that the industry has been focused on in the last half century since the utility of RNA as drug targets have come to light, have some pitfalls to their development, namely low bioavailability and inherent inescapable toxicity, which can be avoided with small molecules. And still, even including, the modern vaccines, small molecules still account for the majority of RNA targeting therapies that have been approved. And excluding vaccines, I think we won't see too much of a shift. The specific RNAs we are targeting with our pilot study are microRNAs. So these are inhibitory post-transcriptional regulators that knock down series of dozens or even hundreds of transcript targets. And so, you can get pathological misexpression of these microRNAs that is inversely proportional to their targets, and they're really attractive drug targets because you can have this innate combinational therapeutic approach. So we have strategies in place for both over and underexpressed microRNA, but our pilot study is looking at underexpressed microRNA 29. MicroRNA 29 has been indicated for Parkinson's disease and pulmonary fibrosis, which are fairly large markets. And because there's such an unmet treatment need, these markets, any therapeutic that is effective would be widely adopted, and these markets would be largely taken. It's been indicated for many other things as well, but we didn't select microRNA 29 because it's a panacea. It was selected because it has evolutionary conservation, so it can be tested in animal models. We can target it with our technology, and there are clear avenues to effective therapeutic approaches and no hazardous targets indicated. And as extra feather in its hat, microRNA 29 as a drug target has already made it to phase 2 clinical trials, where it was ultimately pulled for extremely poor bioavailability. And it also is present in circulating exosomes, which, if you're not aware, are kind of natural lipid nanoparticles that offer a backdoor of bioavailability, able to cross the blood-brain barrier. And expression is also very compartmentalized, so you see expression in the target tissues in the brain and lung, but not in places like the liver. So the proprietary technology we bring to the table is kind of 3 phases. The first is our docking simulations that are RNA compatible. So the state of molecular dynamics in RNA is caught up to where proteins were a decade ago, which is certainly a serviceable state for drug design. The second one is our novel novel small molecule generating engine where we can select for bioavailability and specificity. Specificity such that single nucleotides aren't targeted by the small molecules we make. And the first part, at least, the docking strategy has now been tested in vitro, at least, using commercially available tool compounds. And we are looking to raise funding to synthesize as many of the novel small molecules as possible for testing. The most company out there is Expansion Therapeutics. It was founded by Matthew Disney, who has written most of the literature out there about targeting microRNA with small molecules. They use a top-down biophysical approach, or a shotgun approach instead of a designed approach. And their technology is only applicable to overexpressed microRNA. So they're in a league of their own against the other oligonucleotide-based therapeutics that have made it to clinical trial. All of these are dwarfed in safety and bioavailability compared to what's possible with small molecules. And some have slightly higher bioavailability from doing things like being packed in lipid nanoparticles or being conjugated to things with better bioavailability than RNA, but they're still dwarfed by what's possible with small molecules. And some have particularly low safety and had to have trials pulled, which might have been avoided if they used, a more designed, a more bottom-up approach to finding a target. You would have found these hazardous targets, off targets that are hit. So to date, we've designed a great number of hit-to-lead molecules with this exquisite specificity, and with a little bit of non-dilutive funding and a couple of customers, we've been able to, validate the docking platform and initiate synthesis for a couple of hits. And we're looking to raise funding to synthesize more, and we have pipelines in place for both the Parkinson's stream and pulmonary fibrosis streams, and are hoping to get some allies, which could include access to synthetic pipelines. So thank you very much to the boots on the ground. I think Avid's probably here today, all young upstarts with their hands in the lab, and some other familiar faces, key partners that have given some good advice and potential collaboration in the development pipeline. Thank you.
Thank you, Brock. Very interesting and certainly exciting technology and science for sure. Our next presenter is Damien Kettlewell, co-founder and CEO and director of Clairvoyant Therapeutics. Damien. Oh, you're on mute.
Thank you very much, Wendy. Share my screen. Okay.
Yep, perfect.
Here. Good day, everybody. My name is Damien Kettlewell. I'm the CEO and co-founder of Clairvoyant Therapeutics, a Vancouver-based clinical stage biotech company focused on bringing psilocybin therapy to market for alcohol use disorder. Thank you to Life Science Best in BC. It's our second year here, and excited to share some significant progress. So our purpose is to provide evidence-based psychedelic therapy to help patients live healthier and enriched lives. We are operating a phase 2b randomized controlled clinical trial for alcohol use disorder using psilocybin therapy. And as some of you may know, psilocybin is the active ingredient in about 120 species of mushrooms. And so we're bringing it to market with a synthetic form. We have a very strong team with a proven clinical track record, experience, and deep drug development. And we are the most advanced company globally developing psilocybin therapy for the treatment of alcohol use disorder. And we're looking, our target markets are Europe, Canada, and the UK. And we believe, and we're on target to start servicing and marketing this product in 2026. So as per the below final bullet there, we are placebo-controlled, randomized, double-blind. We have 15 sites. Actually, today I received confirmation about 30 minutes ago that our first patient was randomized. So we're quite excited about that. And so the problem, alcohol use disorder, the reason why we chose this indication was for multiple fold, but really it was the large unmet medical need. There are no strong and doctors and emergency room doctors and addiction physicians are quite frustrated. There's no strong drugs or therapy out there currently with any efficacy of less than 25%, that being naltrexone and acamprosate. And even Alcoholics Anonymous, unfortunately, only has an efficacy rate of 9 to 11%. So there's a very large market. Those in our 3 target jurisdictions of the EU, UK, and Canada. There's 339 million people that drink, and about 1% of those are currently seeking treatment for alcohol use disorder. And so we founded our study based on our academic study that was done in 2014, and showed very strong efficacy. But it is a bit of a unique industry because there was a significant amount of prior art. And so in our case, psilocybin, it was synthesized in 1962. And the other classic psychedelics like LSD and DMT and MDMA, mescaline, they're all synthesized going back 60 to over 100 years ago. And so the patents in these patent claims in classic psychedelics, we do not believe will stand up. And so we are focused on what's called data exclusivity. We'll get into that in a little bit. So here it is here. The first company to bring psilocybin to register as a therapeutic product will get 10 years of data exclusivity in the EU and the UK and 8 years in Canada. We see the main competitors as generic drug companies, and the first company to register this product as a therapeutic product will get this data exclusivity. So and be able to keep out generic drug companies. So you can see the timelines there, anticipating considerable sales from 2026 to 2036 until generics come in. The industry has received, there's some very promising studies that have been done, sponsored going back 20 years now through Johns Hopkins primarily. And Morgan Stanley has come out recently and commented on the seismic shift that psychedelic will bring to many mental health and addiction indications. And in regards to our trial, we'll have 9 clinical sites in Finland. We are approved in Finland, and then 6 in Canada. So we presented in November last year. We got approved in Canada to operate. We initiated our first trial site in June this year in Canada, and then about 3 weeks ago, we were approved in Finland. So things are moving in the right direction. We'll have, and we've chosen these Scandinavian countries. I mentioned we chose alcohol use disorder because we believe it has a very quantitative primary endpoint. So we're focused on the reduction of the number of heavy drinking days. And we're piggybacking on some prior work by a non-psychedelic company, a pharmaceutical company called Adial, that that ran trials, and there's a significant clinical base and very qualified principal investigators, site principal investigators, and most importantly, there's 600 pre-screened patients left from that trial, this phase 3 trial. So you can see at the bottom bullet here that we are doing 2 doses of synthetic psilocybin over an 8-week treatment period. And so that it combined with 20 hours of psychotherapy. So it is a unique modality with drug administration and therapy. So there's 2 1-hour sessions of 2 1-hour motivational enhancement therapy session, and then an 8-hour session is actually occurring today with 2 therapists in the room for 8 hours, 1 trained in psychedelic specific therapy and 1 trained in motivational enhancement therapy. And then a 3rd session, integration session after the first dosing occurs 4 weeks after the first dose. And fortunately, our strategy is we're grateful that it is moving in the right direction. You can see a study here that was, I mentioned the 2000 at the University of Mexico that showed a 60% drop in the number of heavy drinking days. And so that principal investigator moved to NYU Langone. And some of you may have seen in JAMA Psychiatry in August this year that this follow-up study was done with 95 participants that received 2 doses of psilocybin with therapeutic counseling. And you can see the significant drop in the 4-week intake period. So there's 4 weeks of screening. And so the therapy alone, the brown line is the therapy alone. And you can see that the placebo alone with therapy has considerable drop in the number of drinking days, but in the active group with psilocybin, a significant reduction on the psilocybin group, more so than the placebo group. So this is very promising data, and our trial design is very similar, almost exactly similar to this trial design. And so our investors are quite happy that our clinical program is being de-risked in regards to efficacy. So we have in the last year been getting the word out. We're a private company, but we have been getting some media here and there and getting the word out about Clairvoyant. And then in regards to revenue, we believe we can have 3,200. You can see at the bottom bullet point, 3,200 trained psychotherapists, 250 treatment centers, and with 4 treatment centers in each room. And so having 1,000 treatment centers in 2030 alone, just focused on revenue from selling these 2 pills, will get us to $500 million in sales in 2030. And there's going to be partnerships required to protect this data exclusivity. We can get into that for those that are. We look forward to engaging with investors after this today. I'm going off to doing some traveling in the next couple of days. But here we are now in our seed round. So you can see our institutional investors there, institutional psychedelic medicine investors, and now we are doing a bridge financing of $3 million at a pre-money of $12 million, and we do have a term sheet in place with our lead investor. So this is some information on how to of myself and our CFO if you fancy, and we look forward to engaging. And thank you very much again to Life Science BC for the opportunity to speak today. And we look forward to presenting next year at this time and announcing that we are moving into a phase 3 trial, because we believe this is a registration trial, this phase 2B trial, and our next phase 3 we'll be able to do 1 phase 3 trial as our plan, and then register with these 2 trials and get a product to market with 2 trials. So thanks again for your time.
Thanks, Damien. It's great to, as you say, you presented last year. It's great to see the progress and all the optimism as you're looking ahead. So congratulations on your milestones to date. I would now like to invite Stephen Shalmong, VP Capital Development at Microbion, to present.
Okay, thank you so much, Wendy. Just get my screen share set up here. Okay, you able to see everything here?
Yes.
Okay, well, hello everyone. Thank you for the opportunity to present today. My name is Steve Shalong, and I'm very pleased to present Microbion Corporation today. I have been involved with Microbion since launch as a founding board member, advisor, and investor, and today I lead investor relations and our corporate development efforts. Now, Microbion was started after our founder, Dr. Brett Baker, nearly lost his newborn daughter to a life-threatening respiratory tract infection. She was only minutes old when it seemed that her life might end. Thankfully, her life was saved by a broad-spectrum antibiotic. Since that time, Dr. Baker has dedicated his life to solving the growing crisis of antimicrobial resistance, or superbugs, and biofilm-associated diseases. Today, Microbion is a clinical stage company advancing a proprietary new class of therapeutic drugs for the treatment of rare and serious diseases. Our lead drug candidate, pravibismane, is the first drug in a new class designated by the World Health Organization. We are targeting indications with high unmet medical need, which collectively represent an estimated $6 billion in peak annual sales potential for pravibismane in the US alone. We have a clinical stage pipeline with 2 programs in phase 2 development and a 3rd program anticipated to enter phase 1 human trials in 2023. So far, pravibismane has been very safe and well tolerated in 5 clinical studies with over 325 human subjects, and also demonstrated efficacy signals in phase 1b/2a studies. We have backing from key strategic partners, including the Cystic Fibrosis Foundation, CARB-X, NIH, and DoD, amounting to over $21 million in non-dilutive grant awards. And we have a strong and growing patent portfolio with protection to 2041, as well as an additional 10 to 12 years of regulatory exclusivity via orphan drug designation. Now, many of you may be familiar with the recent study that came out that showed that antimicrobial resistant infections were the direct cause of nearly 1.3 million deaths in 2019, and up to 5 million associated deaths globally. By 2050, AMR infections are projected to become the leading cause of death worldwide, according to another major study. What you may not know is that bacterial biofilms are a major contributor to resistance in chronic diseases and are estimated to be involved in up to 80% of human infections, according to the NIH. Biofilms not only provide physical protections for bacterial and other pathogens, but they also enable up to a 1,000-fold increased rate of mutation and upregulated horizontal gene transfer compared to planktonic bacteria. The reality is that most of today's approved therapies are unable to penetrate bacterial biofilms, which inhibits their potency and efficacy. Consequently, treatment of biofilm-associated diseases is an area of very high unmet medical need today. Pravibismane is truly differentiated from all other approved therapies as it demonstrates very broad spectrum potency against a wide range of bacterial and fungal pathogens, including multi-drug resistant pathogens or superbugs, combined with an unprecedented ability to penetrate and eradicate biofilms. Pravibismane has a novel mechanism of action that inhibits bacterial cell membrane potential, resulting in bioenergetic inhibition and a rapid reduction in ATP. Importantly, pravibismane does so without membrane disruption and demonstrates no adverse effects on mammalian cells. And so far, pravibismane has shown low susceptibility to the development of resistance. This unique broad-spectrum potency was recently demonstrated in WHO-sponsored studies in which pravibismane and many other commercially utilized comparator antibiotics were tested against nearly 3,000 contemporary globally sourced pathogens, including multi-drug resistant pathogens. In this heat map, green indicates high potency via very low MIC value, and red indicates weak potency. In the first column on the left, you can see that pravibismane is very green, demonstrating activity against all isolates tested. Furthermore, when compared to the 17 comparator antibiotics, pravibismane was the most broad-spectrum agent evaluated, demonstrating equal or superior potency. Now, this broad-spectrum activity is also complemented by the ability to eradicate biofilms at the same therapeutically achievable concentration as the MIC values. And this dual activity is truly unique, as you can see from a direct quote from Dr. Matthew Parsek, a leading biofilm research expert at University of Washington. We currently have 3 drug development programs underway. Inhaled pravibismane will be entering phase 1 development for the treatment of lung infections. We have completed preclinical studies in which pravibismane demonstrated in vivo efficacy against Pseudomonas aeruginosa and non-tuberculous mycobacteria lung infections, and was consistently well tolerated in 28-day GLP toxicology studies. We anticipate start of phase 1 human studies in the beginning in H1 2023. Local pravibismane is in phase 2 development for the treatment of orthopedic and prosthetic joint infections. And in our phase 2A studies treating orthopedic device-related infections, pravibismane was again very safe and well tolerated at all doses, while also demonstrating a 50% reduction in orthopedic treatment failures compared to placebo. And topical pravibismane is in phase 2 development for the treatment of diabetic foot infections. Here you can see images from our phase 1B studies that were completed, treating moderate and severe DFI. The mean age of these wounds was 6 months when entering the study, meaning that these wounds had not healed using standard of care for 6 months. In the first pictures on the left in this series, you can see the wounds when they entered the study. And then in the second and third pictures, you can see how these wounds continued to close 2 weeks and 4 weeks after end of the pravibismane treatment. While this was a safety study and not powered for efficacy, pravibismane did demonstrate a 3-fold reduction in wound size and 6-fold reduction in amputations compared to standard of care. Microbion has been awarded a $2 million non-dilutive grant from the U.S. Navy to fund a phase 2 proof of concept study treating moderate and severe DFI. All of these indications represent strong commercial potential. Topical treatment of chronic wounds and DFI represents nearly $3 billion in peak annual sales potential for pravibismane in the US alone. Inhaled pravibismane for the treatment of cystic fibrosis lung infections and non-tuberculous mycobacteria lung infections represents $2 billion in peak annual sales potential in the US, and orthopedic infections represents $1 billion in peak annual sales potential in the US. We do have a very strong management team in place that brings over 100 years of collective experience across drug product development, regulatory, CMC, and commercial launch at companies that include Merck and Seprecor. Pravibismane, hopefully you can see, offers a very unique constellation of benefits with strong potential to surpass all other approved therapies. We have also recently discovered a very important new mode of action that extends beyond its known antimicrobial and antibiofilm activity that will either be complementary to our existing indications or open up new opportunities for new indications. We're happy to share more details under confidentiality as we have just filed new IP. We are currently raising capital to advance our current programs to achieve key value-driving milestones over the next 2 years. This includes completion of our phase 2 proof of concept study treating moderate and severe DFI, and completion of our phase 1 study for inhaled pravibismane treatment in healthy volunteers. I invite you to reach out to us to ask questions and discuss how you can get involved with our mission. Thank you so much for your time and interest today.
Thanks, Steve. Great presentation. Again, an organization that we're quite familiar with, so it's really exciting to see where you're at now and the milestones that have been achieved. So congratulations.
Thanks for the opportunity, Wendy.
Yeah,
we're excited.
Yeah, it's fantastic. Next, we have Julie Resler, the Vice President of Corporate Development from Primary Peptides. Over to you, Julie.
Thank you, Wendy. Okay, great. Good morning, everybody. I'd like to express my thanks to Lumira Ventures, BC Life Sciences BC, and Genome BC for the opportunity to present to you today. It's my privilege to introduce Primary Peptides. We are a protein manipulation company focused on neurological disorders. We're a spin-out from the Centre of Brain Health and the Faculty of Medicine at UBC, and we are a clinical stage company, excuse me, with first-in-class peptide therapeutics focused on RNA drug targets in neuroindications that address major unmet medical needs. Our most advanced asset is K13 for acute stroke, and we have positive phase 1 data. Our second asset is PP03, indicated for Parkinson's, and our third is PP07, indicated for ALS or Lou Gehrig's disease. And both of these preclinical assets are ready to enter IND-enabling studies. We have a strong patent portfolio, and we actively seek strategic collaborations with our candidates and platform. And we're here today seeking our Series A financing for a $10 million investment to advance our pipeline and our platform. So what is the problem? Most drug targets are proteins, but most proteins cannot be drugged using conventional methods, which really are small molecules and antibodies. Only 15% of the human drug to date using these conventional methods. The remaining 85% is considered undruggable or yet to be drugged. And these proteins do not have the correct features or druggable binding pockets to enable them to be inhibited by small molecules or antibodies. Intractable proteins present a key challenge in modern drug development, requiring innovation and leading new technologies. So our solution is to expand the pool of druggable targets using and proprietary technologies. The first is protein-protein interference, and the second is targeted protein degradation. And we peptides. We do not employ small molecules or antibodies. Our advantage is our ability to precisely identify the exact contact points of any arbitrary pair of proteins. If you look on the diagram, if you look to the left, you can see a yellow protein and a green protein. And the green neon stick figure is our therapeutic peptide. We have the ability to be able to identify the exact points of this yellow protein with its interaction with the green protein. And our green neon stick figure blocks that interaction. These proteins are actually featureless and don't have very observable binding pockets. And so that is the secret sauce that we are able to bring to this technology, is that we are able to identify the exact contact points to block interaction. Our second technology is described to the right of the slide, and we are focused on protein degradation, where we harness the natural degradation machineries of the cell to degrade disease-causing proteins. This is our therapeutic degradation peptide. It consists of 3 domains. The cell-penetrating domain that enables the peptide to cross into the cell cytoplasm and, importantly, cross the blood-brain barrier. The protein-binding domain that seeks causing protein and forms a peptide-protein complex. And then finally, the lysosomal proteasomal degradation targeting motif that then traffics the peptide protein complex either to the lysosome or to the proteasome for degradation. And we conduct experiments to determine which is the better disposal system to degrade the disease-causing protein. Classified within the broad family of PROTAC technology, and PROTAC companies can only degrade disease-causing proteins via the proteasome, whereas we have the flexibility to degrade using either the lysosome or the proteasome. And all our drugs are developed using rational design. So why do we focus on peptides? Well, first of all, they're composed of natural amino acids, so they're naturally occurring within our body. They demonstrate good efficacy, good safety, low toxicity, very few off-target effects, and they are non-immunogenic. And they have very high selectivity with inexpensive manufacturing. However, peptides are known to have a very short half-life, and we are currently working with companies to extend the half-life of our peptides from once-a-day administration to either once a week or once a month. Now, peptide therapeutics are actually well known. Insulin, which was approved almost a century ago, was actually the first peptide therapeutic ever to reach patients. And there are several blockbuster peptide therapies on the market today, including Lilly's tirzepatide and Novo Nordisk's semaglutide, both indicated for type 2 diabetes, but Novo Nordisk semaglutide is also indicated for obesity. And the market is valued at $42 billion with a 10% CAGR. This is our pipeline. K13 is our most advanced asset. It's an interference peptide that blocks the interaction between 2 important targets in acute stroke, P10 and NED4. It's currently partnered with Yabao Pharmaceuticals for greater China rights available, and it has the potential to enter phase 2 clinical studies based on a filing of an IND with the US FDA. Our P is a bifunctional degrader, which means it blocks a protein signaling pathway in Parkinson's disease, and it degrades the disease-causing protein, which is alpha-synuclein. and that is ready to enter IND-enabling studies. And our final candidate is PP07, which is indicated for ALS, and this is also an interference peptide, and it blocks the interaction between TDP43 and a key signaling protein. So we are designing disease-modifying first-in-class peptide therapeutics for major unmet medical needs. TPA is the current standard of care for acute stroke. It is a clot-busting drug, so it's not disease-modifying, and it's only indicated for ischemic stroke, and it has to be delivered within 3 hours after a CT scan confirms diagnosis. K13, on the other hand, our asset, is a neuroprotective candidate. It is suitable for both ischemic and hemorrhagic stroke, so it will target a much significant larger market, and it can be delivered up to 6 hours after stroke onset and potentially in an ambulance setting because a CT scan is not required for diagnosis. Parkinson's is very poorly controlled today. The only therapies on the market are for symptom control. There are no disease-modifying therapies. Our asset targets alpha-synuclein, a key hallmark of the disease. Several candidates targeting alpha-synuclein have actually failed in clinical studies because they're unable to cross the blood-brain barrier. Our candidate brain barrier. And ALS, our 7 targets TDP43, again a key hallmark of the disease, which is present in 97% of patients with ALS. The majority of companies today are actually targeting a different pathology, which is present in 3% of the patients. And there's only a handful of companies actually targeting TDP43. 3 are in preclinical and 1 is in discovery. So we're all about the same stage of development. This slide serves to show the excitement around protein degradation. In the last couple of years, there have been significant deals signed between a big pharma and protein degradation company. But suffice to say that the majority of companies focused on protein degradation actually have limited their therapeutic areas to oncology. Primary Peptides is the only company that is fully focused on CNS disorders, and that is because of our deep expertise in this discipline, but also because of our superior. We are seeking a Series A for a 2-year runway, and by the end of 2025, we expect PP07 to have completed a phase 1 study in ALS, and K13, our neuroprotective agent, to enter phase 2, and we would partner that asset. Remaining funds would be used to further develop our longer-acting peptides, as well as to recruit key expertise. Finally, I'd like to close with a slide showing our accomplished team. We have a proven leadership team with deep expertise in neuroscience and the pharmaceutical sector, and we have an esteemed advisory board with the likes of Ron Peterson, who has a Lifetime Achievement Award for Alzheimer's research, and Helen Burt, who many of you know, many on this call are familiar with, know Helen very well, who's a proven expert in drug delivery. Opportunity to present to you today. Excuse me. Here are my contact details, and I look forward to receiving your questions in the Q&A chat function.
Thank you, Julie. Great presentation. Always interesting to hear what's happening with your organization. It's nice to see the milestones. Our next presenter is Handel Kim. Hi, Handel, co-founder and CEO of Variational AI.
Thank you very much. My name is Handel Kim. I'm co-founder and CEO of Variational AI. Can you see the screen?
Yep, you're good.
Great, thank you. At Variational AI, we use generative AI to create molecules the world has never seen before to become better medicines. Let me start by setting the stage and talking about AI for drug discovery. This has been a trendy topic for several years now. But let's look at the actual impact on the industry. So on the left, you've seen that, you can see these diagrams that show that 24 AI drug discovery companies generated almost half as many preclinical assets as of all of big pharma in 2021. That's 158 versus 333. The adoption of the first generation of AI for drug discovery is now happening at scale, and it's only increasing. Our proposal is that generative AI, which is the type of AI that we use, will in turn massively accelerate this adoption. It is clear that AI for drug discovery is no longer a fad or a trend, but is actually being adopted and will only increase in adoption. So let me step back and kind of frame the problem. This is a little bit of a glib comic that talks about the streetlight effect. We believe that good drugs start with good molecules, but we're not searching in the right places. We have a tendency to search in places where we can see rather than places where we actually want to find. This is not surprising at all, given the fact that chemical space, where all drug-like molecules come from, is a massive space of 10 to the 60 drug-like molecules. It would be absolutely foolish to blindly look in areas of chemical space, especially because most of it is not full of drug-like molecules and will be useless. So how do you deal with a problem when you have such a massively large space of which we've only explored 0% to date? That is a question that drives us at Variational AI. We ask ourselves, how many life-saving blockbuster drugs are waiting in chemical space to be discovered if only we could discover them? Chemicals, it's too big to screen, but that's usually the first step because that's really what is available to us. The first step is obviously to find hits by experimentally or virtually screening libraries of molecules against a selected target. The status quo on high throughput screening, which is experimental, you might get hundreds of thousands or a million if you're a big pharma. It's expensive, it's slow. You can use virtual high throughput screening and computationally accelerate the number of molecules in your library that you're throwing against the target into the billions. And then the first wave of AI, which is based on discriminative models, can in turn massively accelerate this by up to 10,000 fold, such that you're looking at tens of billions of molecules in your library against a target. But at the end of the day, you're throwing the same libraries against different targets. It's difficult to get novelty or diversity outside of that library that you're using. It's clear that library-based screening is not enough, even with first-generation discriminative AI. So I use this term discriminative, and what does that mean? Well, it's a type of model, and AI is not just one method. It is not one omnibus tool that you use across drug discovery. You use the right tool for the right task. What discriminative AI does, you train a discriminative AI model to classify whether a new molecule is a hit or not against the target. Discriminative AI is the type of AI that you would see in medical imaging, in autonomous driving, in many applications today. It's already broadly adopted across multiple industries. Generative AI, on the other hand, is a new form of machine learning, where you're training a model not to classify whether a molecule is a hit or not, but you're training it to actually make brand new molecules that you've never seen before. And generative AI is starting to rapidly disrupt multiple industries. To date, most of AI for drug discovery has relied on discriminative models. And that's because AI has been reduced to practice by domain experts, computational chemists, cheminformaticians, but AI itself is a science. And at Variational AI, we are AI scientists. We're pioneers of generative AI. Our drug discovery platform, which we call Enki, is based on our proprietary generative model and is based on a variational autoencoder framework. What Enki does is it generates novel molecules from unexplored areas of chemical space and optimize novel starting points and developable scaffolds in order to de-risk lead optimization and increase the speed of development. How does it work? Well, you may be familiar with this model called DALL-E. It's kind of taking the world by storm. What you want to do is you want to generate an image. And all you need to do to generate an image is to use text to describe what image you want. On the left, I prompted girl with short black hair rides a red bicycle over. Boom, the generative AI model generates brand new. These are not searched images. These have been created by the AI. In each case, you see that the images are different. But in each case, you have a girl with short black hair smiling, riding a red bicycle over a bridge. The generative AI creates diverse images that satisfies the descriptions. That is exactly how Enki. Instead of describing what we want to see in an image, however, what we do is we input the desired molecule target properties as well as endpoints, and then Enki generates novel yet diverse molecules that fit this description. That is the power of generative AI. We are not searching, we are creating. Does it work? We were able to validate this in a proof of concept on an asset we call VAI 1122. It is a SARS-CoV-2 main protease inhibitor. We were able to generate a number of compounds, get a very good hit rate. We've got 2 series of novel drug-like hits that show selectivity and synthesizability that we're patenting, developing, and moving forward. At the same time, we are not a COVID company. We are, in fact, training the platform. And where we're going next is we're starting with protein kinases. And we're choosing protein kinases as the initial focus for the company because it is well understood. There's a relatively de-risked biology. It's the second most frequent target class for all drugs. It's a big market. It's the largest segment of oncology drugs. And importantly, there's a lot of training data because we are a machine learning company, but also there's a lot of CRO capability. And we don't have to reinvent the wheel with respect to assays and development. What we bring to the table in developing, in going after kinases is a unified kinase model. So instead of training on data for only one kinase target, we are able to train on multiple kinases, many kinases, to create a unified understanding through Enki to be able to rapidly generate novel kinase inhibitors across multiple targets, synergistic targets. Our business model is very simple. We currently have a partner model where we work with a customer that selects a kinase target that has an understanding of the biology. We use the Enki unified kinase model, we generate lead-like hits, and then of course the customer takes forward and develops. We in turn get upfront milestones. We have one customer signed already and more in the funnel. This is lower risk, lower reward path, and it's what we start with. What we're trying to do today is raise a $25 million Series A, where we're going to start working on an internal pipeline. Obviously, it's higher risk, higher reward. But the great thing is that we use the same models that we're training to generate the lead-like hits. We just simply assume more of the risk. In the competitive landscape, there are many companies that are doing AI for drug discovery. However, we are the only ones that are focused on generative AI for kinase inhibitors that have an internal pipeline development model. The team is a good balance of machine learning, cheminformatics, as well as medicinal chemistry. And our SAB and advisors are world-class. We're seeking $25 million Series A in order to fund the development of 5 internal programs in oncology. With $3.5 million US that we raised on our seed, we were able to increase the size of the team and develop our first unified kinase model and sign our first customer. With Series A, we're trying to fund the development of 5 programs and develop a second unified model in a non-kinase target class and license 2 assets. This will fund us until Q4 of 2025. And looking forward in Series B, that's when we really turn the crank and begin executing at scale. My name is Hanel Kim, and I look forward to any questions. Thank you.
Thanks, Handel. Excellent presentation. Another company that we know quite well, and it's really exciting to see what's happening. You're getting lots of chats on your analogies of the girl with the black hair and street lamps. So a great way of describing what it is that you're doing and the impact of it. So with that, I want to thank all of our presenters, Brock Schumann, Damien Kettlewell, Stephen Shalong, Julie Resler, and Handel Kim. All of you had amazing pitches, certainly very interesting. You've set a high bar for the conference as we go through more pitches over the next couple of days. As I mentioned at the beginning, this session was sponsored by Genome British Columbia. And I'd now like to introduce Tony Brooks to say a few words on behalf of Genome BC. Tony's a senior executive with over 25 years of experience in life sciences, not-for-profit, and public accounting environment. He leads Genome BC's finance, HR, administration, and entrepreneurship and commercialization areas. And with that, Tony, thanks very much, and the floor is yours.
Thank you, Wendy. I hope you can all hear me.
Yes, we can.
And I apologize profusely for the technical issues that we had earlier. So now I'm going to do a brief introductory presentation at the wrong end of the session. but that's okay. We'll manage it. So I'm not going to talk for very long. Thank you all very much for attending, and thanks to the other presenters who have given some fantastic presentations. So I'm just going to quickly share the screen here. Hopefully you can see that.
Looks good.
Oops, sorry.
You just went
Back
out.
to there we go.
There you go.
So Genome BC, we've been around for 20 odd years, and you know, we have enjoyed working in this wonderful and vibrant ecosystem as a partner to many people. So why genomics? And I guess first of all, I should point out that we take a very broad definition of the term genomics. It's not just genomics that we work on. We work on proteomics, metabolomics, any kind of omics you want, really. We'd like to keep it pretty broad. But why omics? Because, you know, omics can impact many aspects of our lives, not only human health, but food security, environmental stewardship, climate change and adaptation, just to mention a few. Who we are, our vision is that genomics positively impacts life every single day. And our mission is to apply the power of genomics to pressing societal and economic challenges. And the way we do that is we interface between academia, government, and industry. We work across many different sectors. Some you might find somewhat surprising, the more obvious ones like health and agri-foods. But in addition, forestry, fisheries, aquaculture, environment, mining, you can apply genomics to mining remediation. So we have projects in mining and in energy. We have a record of success, as many of you may know. We've funded over $1.25 billion of research projects, almost 500 projects, over 1,000 partners, almost $1 billion in co-investments. And that has produced over 3,500 scientific papers, over 750 patent applications, and we've helped 152 companies advance. So in addition to that, we have this thing called our I2 fund, or Industry Innovation Fund. And our goal with this fund is to help early stage life sciences companies move across the funding gap that is highlighted in orange on this slide. So essentially, 5 years or so ago, we set up a $20 million evergreen fund. We make loans. So we're a debt fund. We make loans to early stage companies, and I appreciate that. For some people, that's a little bit of a strange concept, that you have a early stage company that will have debt on its books, but that's what we do. We make loans of $500,000 to $1.5 million, and these companies have to have a high potential of commercial success. The loans are repayable within 4 to 6 years. They carry a low interest rate, prime plus 3, and I appreciate in these days that is not as low as it was, say, a couple of years ago, but it's low relative to other organizations that lend. We have success fees in the form of low single-digit royalties based on revenues. So if the company goes on to be successful and generates revenues, there is a royalty, but that royalty is capped at 2 times the loan principal. So the royalty doesn't go on forever; it just caps it. It's capped at 2 times the loan principal. And we have a mechanism for early repayment incentives. So if you pay back before 4 years, you can reduce that royalty cap. So this is a unique model. The model has been going for, as I say, 5 years. It's proving to be very successful. And if you're interested, please come and talk to us. And those are some of the companies that we have partnered with over the years. Looking at this list, you don't actually have to have a company that begins with the letter A or S. That's just a coincidence, but there are lots of A's and S's in that list. And that is my short presentation. Thank you very much to Wendy and her team and to Life Sciences BC for the opportunity. And we really hope you enjoy the other sessions, because as noted, there are some fantastic companies presenting today. So thank you.
Thank you, Tony, and thank you, Genome BC, for sponsoring this session and for your support of the sector generally. You play an incredibly important role in the early stage innovation in the life sciences sector, and as you talked, in much broader than just life sciences, obviously. As we close out this session, a reminder to check out our digital presentation, presenting company guide. It was put in this chat. It's going to be put in the chat of every pitch session, but it's also available in the main stage. In addition, we encourage you to reach out to presenters via the People tab and connect with them. You can chat with them through text, set up video meetings on the platform, and make connections. And that's what this event is all about: of having companies meet investors, but also have innovators meet innovators. We have another Therapeutics session starting up in about 15 minutes, so that's 11 a.m. PST. So hope you'll be able to join us there. And thank you again to our presenters. As I said, you set a very high bar on not only your presentation, but also the amazing innovation that's happening in your respective organizations. Thank you, everyone.